Hey, all right, so this is going to be the fungi lab. So this is what we would have done in class or lab today. We would have taken some mushrooms and cut them up and looked at them. So I'm going to do this virtually with a PowerPoint and talk to you about the structures you need to know about the fungi. There's three types of fungi, so I'm going to go through the three different types and give you some structures for each type. Okay, and that'll be what you need to know for that portion of the lab. So I'm going to take the camera off and pull up the PowerPoint. Somewhere back there. Where'd it go? There we go. Okay, there we go, fungi. It's not as easy as it is to do this in my office. So this is a picture of a strawberry, as you can tell, and it's fuzzy. And so if you've ever wondered what the fuzzy stuff is on a strawberry or an apple or anything, that is the fungus. Okay, and so you probably knew that. A lot of people say mold, but it actually is a fungus, which is a little different than the molds that we talked about earlier that were when we talked about the ones that had to do with algae and plants. So these are actually fungi. And so these little hair-like projections are called mycelia, and they have something to do with reproduction. So when you read about the fungi, pay attention to what the mycelia do. I'll talk a little bit about that today. So there's three types of fungi. The first one are the zygospore. So this is the phylum Zygomycota. And these are saprotropes, which means they are parasites, which will eat on produce and soil produce and worms. And the one that I'm going to focus on is your black bread mold, the one that we've all seen if you leave your bread out too long. So Rhizopus stolonifer. And so it has several different structures I'm going to point out to you. The hyphae and the sporangium are going to be the main ones. Okay. So this is bread mold, and so you can see that here. And so if you, what happens is you have these spores. So this is a sporangium, and this is a blown up version of a sporangium. You can see all these spores that are being released. Well, when these spores are released, they're going to land on a moist surface like bread, and they're going basically to start to grow. And the spores are either male or female. And they start to put down these kind of hair-like structures that look like roots that are either positive or minus, which is basically the same thing as saying male or female. So a plus and a minus have to come together in order for sexual reproduction to occur. So you have the formation of these spores. They put down these quote-unquote roots called mycelium, and these come together. And this is where sexual reproduction comes into place. So you have fertilization. So you have the male and female come together. You get fertilization, and that forms this sporangium that's called a zygospore as it starts to grow. And this zygospore then sends up this little stalk with the actual sporangium right here, and then you release the spores. Okay, so for the lab test, I'll either have a picture that looks something like this, or perhaps a diagram from your textbook with some of these things X'd out. So you'll fill in the fill in the blank for different parts of the zygospore fungi, like bread mold. Here's another picture. So if I said, identify the organism on this bread, on the bread would be the black bread mold. And on this particular picture, you can actually identify the individual sporangium where the spores are going to be released from. So that's what you would identify for that type of fungi. The next kind are the sac fungi, phylum Ascobacota. And so they have a cup-like structure. It's called an ascocarp. And they make spores on something called a conidia. So those are the main anatomy or part names that you're going to be responsible for the sac fungi. I'm going to show you a picture. The sac fungi are an important group because we do eat them. Their morels and truffles are things that are considered delicacies. Truffles are usually found around oak trees or hazelnuts, like by pigs. They dig them up and they're used to flavor cheeses and oils and different French dishes particularly. And the morels, we actually have those here. You can actually find these in the forest and use those as a side dish for a meal. They're actually really good. I've had them in my place in Tennessee. So this is an example of different types of these sac fungi. So this is the ascocarp here. So it looks kind of like an upside down bowl. You can also see it here. And this is a morel. And so the morel really looks more like a sponge, but it's still called an ascocarp. So this is what you're looking for, like in the woods of Alabama and Tennessee this time of year. You can pick those up and eat them. But you got to know what you're doing. There's one that looks just like it that's not a real morel, and it'll make you sick. So you got to know the difference between the true morels and the false morels. But the true morels look like a sponge, and these are all examples. So on the lab test, you'll place a picture of something like this, or the morel, and I'll say what type of fungi it is. This is a sac fungi. I'll say name this bowl-shaped structure or sponge-like structure, and you'll say that's the ascocarp. Okay? Now, fungi can reproduce asexually, and so a great example of that is yeast, the same thing that you use to make your bread and beer. Yeast is a fungi, and it will bud. And so here's the little yeast budding off of the other yeast. So again, on a lab question, I might have this picture. I say maybe identify the organism. It would be yeast. Identify the bud. Okay. And another one is penicillin. Penicillin is a fungi, and it's the one that we use to make penicillin, which is the antibiotic against bacterial infections. And this is a blown up version of the conidia. And so instead of little buds, they form these kind of strands with the spores on those there. So these structures with chains of spores are called conidia. So that would be something else you could identify. So I can have any one of these pictures on the lab test and then say name a structure. So a good thing to do would be to take these pictures and X out the words and then be able to go through and identify them. And the last type of fungi is the club fungi. This is what you probably call a mushroom. So these are the ones that we think of when we think of a mushroom as a fungus. And so they have something called a basidium with a basidiocarp. So those are going to be the things that we look at on the next slide. A lot of mushrooms are edible, but some of them are not. Some of them can actually kill you. The one known as the death cap is responsible for almost 90% of the fatalities of people that eat mushrooms. So don't go out and hunt mushrooms unless you know what you're doing, because a lot of them will make you very, very sick. So this is your typical mushroom, which is, again, a club fungi. And so you've got what we call the mushroom cap, which is called the basidiocarp. Okay. Then you've got this, the stem is actually called the stipe. And then underneath the basidiocarp, you have these gills. Okay, and I've got another picture. Whoops, I'm going to show you in just a second. So mushrooms, just like the other fungi, they have male and female mycelium underneath the ground that will allow them to connect for sexual reproduction and then produce these spores, which are released for more mushrooms to be grown. All right. So this is a picture of a mushroom, and so the big mushroom cap is the basidiocarp. The stem is called the stipe, and these are the gills. Okay, so that's the mushroom lab. So if you have a mushroom at home in your kitchen, you might want to cut into it and look at it. And again, I'm going to make a list of structures so that you can have a check-off list of everything you need to know. But there's your lab video for the fungi. See you later. Or not. Bye.